Hello, welcome to CRPS Contender, my complex regional pain syndrome education channel. Today's video is Disrupted Brain Relations. It is part 1 of a 2-part breakdown, as well as a third video which is a simplified version. This is another step down our journal journey where we will be discussing the paper Abnormal Gray-White Matter Interactions in Emotional and Autonomic Regions. This is a very complex paper. If it is too complicated for you, please check the simplified version. I've got pictures in that one. I discuss a lot more about the brain regions that are being discussed. I might give vague references in this one, but I'm not going to get too much into that, though I will be getting more into the testing method that they utilize, which I kind of glossed over in the simplified version because of its complexity. So, about this paper. It was published November 25, 2008 in Neuron by a group of researchers from Northwestern University and University of Toledo. Let's take from the abstract. Chronic complex regional pain syndrome is a debilitating pain condition accompanied by autonomic abnormalities. We investigated gray matter morphometry, which is the structural measures of shape and volume of the gray matter, and white matter anisotropy, which is measuring connectivity via water molecule diffusion direction, in CRPS patients and matched controls. Patients exhibited a disrupted relationship between white matter anisotropy and whole brain gray matter volume. Gray matter atrophy in a single cluster encompassing the right insula, right ventromedial prefrontal cortex, and right nucleus accumbens. A decrease in fractional anisotropy in the left cingulum callosal bundle, and reorganization of white matter connectivity in these regions was characterized by branching pattern alterations, as well as increased and decreased connectivity. While regional atrophy differentially related to pain intensity and duration, the strength of connectivity between specific atrophied regions related to anxiety. These abnormalities encompass emotional, autonomic, and pain perception regions, implying that they likely play a critical role in the global clinical picture of CRPS. So why did the researchers do this study? CRPS develops idiopathically in less than 5% of limb traumas. But because efforts to identify specific peripheral mechanisms for the condition have been unsuccessful, research has shifted to central mechanisms. CRPS patients exhibit abnormal or shifted brain activity to motor tasks, imagined motor tasks, and tactile stimuli, and also show the extent of observed functional reorganizations are linked to properties of CRPS pain. CRPS patients engage regions seen in acute pain and also prefrontal cortical regions. They exhibit blunted emotional decision-making abilities and have lower perceptual learning aptitude. This study attempted to interrelate changes in gray matter to the reorganization of connectivity of white matter using diffusion tensor magnetic resonance imaging, known as DTI, and voxel-based morphometry. CRPS patients' performance on emotional decision-making tasks is far worse than that of chronic back patients, and matches impairments in patients with ventromedial prefrontal cortex lesions. The authors hypothesize that CRPS is characterized by gray matter atrophy in the ventromedial prefrontal cortex. Cortical regions modulating autonomic outflow should also exhibit morphological abnormalities, implicating the ventromedial prefrontal cortex. This area is a region for binding together large-scale networks that process emotional processing, decision-making, memory, your self-perception, and social cognition in general. The anterior insula. This is not a very well understood portion of the brain, but it is super important. We're not sure everything that it does, but we know for sure it does things like sensory motor processing, socio-emotional processing, and attention and salience and speech. This is also your disgust center, whether that is intestinal disgust due to rotten food or moral disgust. And the dorsal anterior cingulate, which deals with emotional expression, attention, and mood regulation. It's part of the limbic system. We are also going to be talking about the nucleus accumbens a lot, which has to do with reward functions and the interface between motivation and action. Using probabilistic tractography derived from DTI, one can study connectivity for brain gray matter regions showing abnormalities in density, or white matter anisotropy, and link abnormalities in gray matter or in myelination to brain connectivity. Now let's first talk about where there were not differences in the CRPS patients compared to the controls. There was no difference in neocortical gray matter volume. There was also no difference in the size of the lateral ventricles between age and sex-matched control subjects and CRPS patients. Whole brain skeletal fractal anisotropy, a global measure of water diffusion over white matter tracts that reflects the integrity of white matter in parallel and perpendicular diffusivities, did not differ between CRPS and match-controlled subjects. Let's talk for just a moment about fractal anisotropy because of how they're using this. We're going to be talking about it a lot, and you need to kind of understand how it functions. So think of a neuron. The axon is the long, skinny part of it. Kind of looks like a bit of a rat tail. That axon is wrapped in myelin sheathing. Water can flow parallel to the axon, or it can flow perpendicular to the axon. When they measure how the water molecules flow in parallel, they can measure damage to the axon. When they measure how the water flows perpendicularly, they can measure damage to the myelin sheathing. And remember, the myelin sheathing is what allows the signals to move quickly, and then the axon is what actually carries the signals themselves. Whole brain skeletal fractal anisotropy showed a significant positive correlation with whole brain gray matter volume in healthy subjects, but not in CRPS patients. This finding can be interpreted as evidence for a general disruption of the gray-white matter structural relationship in CRPS subjects as compared with healthy subjects. Let's specifically start talking about the right medial prefrontal cortex and the anterior insula. A single cluster encompassing the right anterior insula, orbital portion of the ventral medial prefrontal cortex, and extending posteriorly into the nucleus accumbens, exhibited decreased gray matter density in CRPS patients relative to matched healthy controls. The peak atrophy was located in the anterior insula. Gray matter density values, extracted from the whole cluster, were significantly different between the groups and showed a negative correlation with pain duration in CRPS. Multiple regression of gray matter density to pain duration using age and gender as covariates indicated that pain duration was the only significant factor and that its effect was 4 times larger than aging within this cluster. When the cluster was subdivided into underlying anatomical regions, the authors found a significant negative correlation between gray matter density in the right ventromedial prefrontal cortex and the interactions between pain duration and pain intensity, and a negative correlation between the right anterior insula, gray matter density, and pain duration. The results showed that the same right anterior insula and ventromedial prefrontal cortex region exhibited a decrease in gray matter density regardless of sidedness. White matter is also decreased in the left cingulum bundle. Stringent statistical thresholding showed that CRPS patients have a far lower fractal anisotropy value in a cluster within the left callosal fiber tract, located just to the side of the anterior cingulate cortex. The opposite contrast, searching for increased fractal anisotropy, was empty. They did not find a positive result. In the area showing decreases in fractal anisotropy in CRPS, the mean parallel diffusivity was significantly larger in healthy subjects, whereas the mean perpendicular diffusivity was significantly less in healthy subjects. Therefore, water diffusion in this white matter cluster in the CRPS brain increased locally but decreased over the principal fiber direction. And to translate that means that they found that there was damage both to the axon and to the myelin sheathing. The authors calculated, for each group and for each hemisphere, the mean number of connections as a function of distance from a given initial region of interest. This is called a seed. They also calculated the cumulative sum of connections as a function of distance from the seed to measure the fractal dimension of the branching patterns. These are detailed patterns that look the same at any scale and repeat themselves over and over. And fractal branching is what allows neurons, axons, and dendrites to communicate with each other. So you want lots of space filled. That means there's more connections and more ability to communicate between different gray matter subsections of the brain. In CRPS, there seemed to be a prominent decrease in the probability of connections to the posterior cingulate area. This is a long-range connection from the white matter region with decreased fractal anisotropy and is most likely part of the cingulum bundle. In the left hemisphere, the average number of connections at long distances, as well as the total number of connections, were significantly lower in CRPS patients. In the left hemisphere, the mean fractal dimension was significantly less in CRPS, and the scaling range was significantly longer. These findings indicate that the structure of this tract is disrupted even in short distances, and the branching pattern was less space-filling in CRPS subjects. However, no significant differences were found in these measures in the right hemisphere of the brain. Decreases in gray matter density can be due to a loss of tissue from neurodegeneration, which is cell death, or from volume shrinkage without cell death. However, loss of gray matter density with concurrent loss of white matter connectivity would provide further evidence for the neurodegeneration hypothesis. The authors therefore studied white matter connectivity for gray matter areas showing decreased density in CRPS. Using the portion of the right ventromedial prefrontal cortex exhibiting decreased gray matter density as the seed, the authors identified tracts in the callosal bundle, the inferior frontal occipital fasciculi, and the longitudinal fasciculi, in addition to the parts of the uncinate fasciculus and fornix connections. In CRPS, long-distant tracts ipsilateral to the seed, on the same side as the seed, showed reduced probability for connections, especially in the fronto-occipital and longitudinal fasciculi, resulting in reduced total number of connections. Now, there was a lot of different brain regions in there. For the sake of time, I'm not going to say what each of those regions do. I do go into that a bit more detail in the simplified version, though, if you want to check that one out, or go ahead and maybe you already know, or feel free to do some additional research on your own there. These slides are all accessible in my note sheet, so you can pull these up yourself without having to pause the video if you'd like. In CRPS, connections from the ventromedial prefrontal cortex seed were higher to the whole insula and the seed anterior insula, lower to the basal ganglia, which does motor control, and unchanged to the thalamus, which is where most of the sensory input is coming from, primary somatosensory cortex, visual cortex, and nucleus accumbens seed. The authors performed the same type of analysis for the right anterior insula region, showing reduced gray matter density. Unlike the ventromedial prefrontal cortex seed, the anterior insula seed had little contralateral connectivity, opposite side. On the same side, the most represented white matter bundles included the inferior frontal occipital and longitudinal fasciculi, the uncinate fasciculus, and parts of the superior longitudinal fasciculus. In CRPS, the same-sided total number of connections was significantly less, although around 100 millimeters from the seed there was a prominent increase in connectivity. Moreover, the fractal dimension was significantly decreased in CRPS patients, implying reduced space filling. Seed to gray matter target connectivity indicated decreased connections only to the basal ganglion and the nucleus accumbens seed. The authors performed seed-to-seed connectivity analysis between the right ventromedial prefrontal cortex, anterior insula, and nucleus accumbens gray matter regions that showed reduced density in CRPS. In CRPS, connection counts from the right anterior insula to the ventromedial prefrontal cortex were increased, while those from the right nucleus accumbens to the anterior insula were decreased. In addition, the authors found that connection count from the right ventromedial prefrontal cortex to the nucleus accumbens was significantly positively correlated with two separate anxiety scores in CRPS patients. We're going to stop there for the sake of time. We will continue on with the real-world impacts of that in the next video. I know that was a lot of very long brain regions that I didn't really get into, so if you would like a further breakdown of that, please see my simplified version. Hopefully that one will make more sense. There's also images in that one if you'd like to know where in the brain those areas are. Okay, thank you very much for watching. If you'd like to help the algorithm engage with my channel, please like, comment, or subscribe. If you'd like to contribute to my living expenses, my Patreon is CRPS Contender. Thank you very much, Emily Malcontent. If you'd like to see the paper or the notes that I used for this, please check the link in the description. There will also be a link to the slides in that source. Thank you very much for watching. I hope you learned something, and I hope to see ya in part 2.